Hello everyone!
My name is Melissa Eklesia.
My name is Shafia Misha.
And we are from
SMA Negeri 1 Palembang.
In this video, we will present to you our research about
analysis of chlorine content in glutinous rice using Morse argentometry method and efforts to reduce it with ketapang or Terminalia catapa L leaves extract.
The condition of Palembang River is now having apprehensive cleanliness. Wastewater from various community activities and high levels of chlorine and phosphate are one of the damage to the Musi River. According to Malik Adil Nawas 2018, glutinous rice or Oryza sativa alpha glutinosa is a type of rice that commonly cultivated in Thailand, Cambodia, Laos, Myanmar, Indonesia, and several other Asian countries.
Lately, many chemical materials that mix into food ingredients. One of them is chlorine that mixes into glutinous rice. According to Darniadi in Wongkar 2014, chlorine is a chemical material that is generally used as a germ killer. Chlorine will react with water to form hypochlorous acid, which is known to damage cells in the body. In this research, to reduce the chlorine content in glutinous rice, we will use ketapang or Terminalia catapa L leaves extract. According to Purwani R.S.K. Tavani 2013, ketapang leaves contain medical compounds such as alkaloid, flavonoid, triterpenoid or steroid, resin, tannin, and saponin. Usually, ketapang leaves used to neutralize pH of the water in betta fish aquarium or BDAM water that contain chlorine. So therefore, this research purposes to know if ketapang leaves also can reduce the chlorine content in glutinous rice, and this research also purposes to know if ketapang leaves can also reduce the river or environmental pollution from the waste of chemical material, especially chlorine from glutinous rice waste. This research belongs to the type of descriptive survey with qualitative and quantitative analysis method using Morse argentometry method to determine the amount of chlorine contained in glutinous rice. Argentometry titration is a titration using silver nitrate as a titrant, which will form a silver salt that is hard to dissolve. This method was chosen because the compound that will be analyzed is a halogenide group, so it requires the presence of precipitate as the end result of the titration. To make the ketapang or Terminalia catapa L leaves extract, put the water into a pan and heat it. Also put the ketapang leaves into it and boil the ketapang leaves until the water becomes cloudy or brown to black color. Put the ketapang leaves extract into a beaker glass. Prepare the glutinous rice sample and the filtrate. Finally, add a few drops of ketapang leaves extract into the glutinous rice filtrate that containing chlorine. To check if there is a chlorine or not in the sample, the meshed rice will be carefully weighed as much as 10 grams. After that, add 50 ml of distilled water and stir it. Take the filtrate with the filter paper and in each test tube, add 5 ml of each sample filtrate. Add a few drops of potassium chromate and silver nitrate. If the result is positive, it will form a brownish red or brick red precipitate. And if the result is negative, it will form a yellowish to white solution. In each new test tube, add 5 ml of the sample filtrate again. Add a few drops of 10% potassium iodide and 1% starch. If the result is positive, it will form a blue solution. And if the result is negative, it will form a white solution. If from the two tests the result is positive, then that sample is positive containing chlorine.
And the formula that we use to calculate the chlorine content in glutinous rice is first, to calculate the milliequivalent OCl mean is silver nitrate volume multiplied by silver nitrate normality. And to calculate weight of an AOCl in sample is molecular weight of an AOCl multiplied by OCl mean. And this is the result of the calculation from the formulas. And for the data analysis technique that we use is descriptive statistic technique that are used to analyze data by describing data that has been collected as it is without intending to make generalizations. That's why the glutinous rice variable will be analyzed descriptively and present in tabular form.
So from our research, we can conclude that 1. 2 out of 10 sample of glutinous rice from 4 different markets in Palembang positive containing chlorine, and 2. Ketapang or Terminalia catapa L leaves extract can reduce the chlorine content in glutinous rice.
And for future work, researchers hope that this research can be applied as an alternative and natural medicine. For further research, researchers suggest to check and reduce the chlorine levels not only on glutinous rice but other food ingredients.